Hi, everybody. My name is Ted Price. I'm the director of the Center for Advanced Pain Studies at the University of Texas at Dallas. I've been an ISP member since I was a graduate student way back in 1999 is when I joined ISP. I've spent basically my entire career in the pain field trying to better understand why people develop chronic pain and to develop better therapeutics, non-opioid therapeutics to treat chronic pain. In the last several years, we have taken a slightly different road to our research. When I first started doing work, we primarily worked with preclinical animal models. But in the last 5 years, we've really dedicated almost all of our work to doing what I call human molecular neuroscience. So the idea behind this work is really to gain a better understanding of the pain system in humans, and we've focused most of our work on the peripheral nervous system, focusing on a particular set of neurons called nociceptors. These are the neurons that innervate essentially your entire body, and they're the start of the pain pathway. When you have damage to your tissue, these neurons become active, and we know that in many people that have chronic pain disorders, in particular neuropathic pain, these neurons become hyperactive. They start to produce what's called spontaneous activity, and most of the evidence that we have suggests that this is a really key driver of neuropathic pain. So why have we switched our work to trying to study the human nervous system rather than using animal models? Well, I think what we've done is identified that there's a really important gap in knowledge and how we understand how we should develop therapeutics that hopefully will be very efficacious in people with chronic pain. So because we know a lot about the peripheral nervous system and nociceptors in animals, but not a ton about the molecular makeup in humans, we've really strived to fill this gap. And to do so, we've done a couple of things. One of them is to work with organ donor networks, so specifically the Southwest Transplant Alliance, which is the second biggest organ procurement organization in the United States, and also to develop collaborations with neurosurgeons that do surgeries that are relatively rare, but where part of the peripheral nervous system is removed from the patient during the surgery. Collectively, what these resources have allowed us to do is to use single and spatial RNA sequencing technologies to really gain amazing molecular insight into the makeup of human nociceptors. And because we are now able to get really detailed medical information about the organ donors and also to work with patients that are having these relatively rare surgeries where part of the nervous system is removed, we've also recovered many samples from people who either died with a chronic pain disorder or have an active chronic pain disorder and are having a surgery. What we've really done over the last few years is to gain really profound new insight into the molecular makeup of human nociceptors and how they change when people have chronic pain disorders. Our hope is really that this kind of very detailed molecular information will lead to the identification of new therapeutic targets that will have really good efficacy for people with chronic pain. And really the goal is to better understand the underlying mechanisms that cause individuals to have chronic pain and try to reverse that with what we call disease-modifying therapeutics. So ultimately, that is what my laboratory is focused on pretty much every day now. Over the last several years, we've built up these resources to be able to do this kind of work. And collectively, I think our lab and many other labs that are also doing this kind of work, such as Rob Giroux's lab at WashU and Will Renthal's lab at Harvard Medical School, are really pushing the field forward in new and exciting ways that I think are going to have a profound impact on how we think about treating chronic pain and hopefully eventually lead to cures for chronic pain.